Welcome to this video where I look at the structure and function of enzymes and also how they are regulated. So what are enzymes and how do they control the rate of reaction? Well, enzymes can increase the rate of reaction by lowering the activation energy, and this is the minimum energy required to break bonds in the reactants in order to initiate the reaction. Enzymes are said to have a compact globular shape. Each enzyme has a region called the active site, and this is where the substrate binds. And the substrate is the molecule or molecules that the enzyme facilitates a reaction through. So here we have an enzyme active site and we have a substrate molecule. And in this case, there is one substrate molecule. The substrate molecule binds to the enzyme active site, and you can see they are complementary in shape. And this is called the enzyme substrate complex. Okay. Now, here, the enzyme substrate complex, you can see now this molecule has been broken down into two separate molecules, the products. The products have a very low affinity for the enzyme active site, whereas the substrate has a high affinity for the active site. So the products are dissociated and they are released. So this is an overview of how an enzyme catalyzes a reaction. Here it's worth noting that some enzymes can bind to more than one substrate and allow them to react together. Some enzymes, like digestive enzymes, as shown in this example, bind to a substance and break it down into smaller molecules. How do mutations affect enzymes? Well, firstly, we need to understand what mutations are. Mutations can be a change in one codon or more than one codon. Now, if we have a codon, let's say CTG, if we change the last nucleotide in the codon, this will give rise to a different codon. This may or may not change the amino acid that this codon codes for. This is known as degeneracy. Now, changing one nucleotide in a codon has a potential to change one amino acid in a polypeptide chain, no more than that. But we must remember that it may mean that the same amino acid is coded for. But in general, changing the codon sequence will alter, that is in the DNA, will alter the amino acid sequence in the polypeptide chain. And because of this, the new amino acid that is introduced will have a different R group, and this may alter hydrogen bonding or ionic interactions or hydrophobic interactions with other amino acids in that polypeptide. This will change the tertiary structure, and this will result in the change in the shape of the active site. Now, when we change the shape of the active site, this may mean that the active site may bind to a different substrate. So it has a different specificity. It may mean it might have a weaker binding to its existing substrate, which will mean the activity will be much reduced compared to a normal enzyme, or it may not bind at all, which means the enzyme is non-functional. These are some scenarios. It's also possible that you might have stronger binding to the existing substrate, which means that perhaps the activity might be higher. There are two general theories that we learn about how enzyme active sites bind to substrates. The first one is the lock and key theory. Here you can see an enzyme E and a substrate S. And you can see the shape of the active site of the enzyme is exactly complementary to the shape of the substrate. So you get an enzyme substrate complex here. This suggests that a particular enzyme active site will only bind to one specific substrate and no other types of substrates. So this is a highly specific interaction. Another model, known as induced fit, stipulates that the enzyme here has an active site which is complementary to the substrate, but it's not an exact match. So when the substrate binds to the enzyme, this alters the shape of the active site slightly for a more precise fit, and this is an induced fit. Now, there's evidence for induced fit, including the idea that inhibitor molecules can bind to the active site where the substrate binds, and this observation supports the induced fit theory. So in summary, lock and key theory states that the substrate shape is exactly complementary to the shape of the enzyme active site. The induced fit theory states an enzyme active site is similar in shape to the substrate. Substrate binding changes the active site shape for a more precise fit. Now let's look at enzyme inhibition. And I mentioned earlier that the induced fit theory suggests that maybe substrates or molecules which are similar in shape may bind to the same active site, inducing a more precise fit when they bind. The lock and key theory is less supportive of the idea of more than one type of molecule binding the active site. So substrate binding to the active sites is inhibited in the types of inhibition that we're going to look at. And the types we're going to look at is going to be called competitive. So competitive inhibition and also non-competitive. There are other types of inhibition, but the ones we're going to look at involve inhibition or prevention of substrate binding to the active site. And both of these types of inhibition are reversible. That means the inhibition can be reversed. Okay, let's look at non-competitive inhibition then, which may be something that you may initially find more tricky to understand, but I'm starting with it because it will give us a better idea of what's going on. So let's have a look at our enzyme here. Let's say I've got 4 enzyme molecules. They're the same enzyme. They have an active site with this V shape. 2 of the enzyme molecules are bound by a substrate. Okay. Now, if I introduce, these enzymes here on the right hand side could also bind the substrate. But if I introduce a non-competitive inhibitor, this inhibitor will bind away from the enzyme active site, and this site is sometimes called the allosteric site. And what will happen when it binds, this active site will change shape. So what will happen is, let's say that the active site shape is now like this. So I'm going to bind the non-competitive inhibitor, and the active site shape is going to change. And now these enzymes can no longer bind to the substrate. The substrates won't fit. There may be plenty of substrate available, but what will happen is when the non-competitive inhibitor binds, this will reduce the number of enzymes which are able to bind to the substrate. It will reduce the number of active sites that are available to bind to the substrate. And this will lower the activity. And this is a very important consideration. How do you reverse this non-competitive inhibition? Well, by removing the non-competitive inhibitor. The non-competitive inhibitor doesn't bind permanently, it can dissociate. If you reduce the concentration of this, it will reduce and eventually eliminate this type of inhibition. We're looking at the curve here of the initial rate of reaction and the substrate concentration. Now here, when we change or increase the concentration of substrate, what will happen is that firstly, at lower substrate concentrations, most of the active sites will not be bound by substrate. If I now increase the substrate concentration, more active sites will be bound. However, if I now have, if I remove this enzyme and have it in the shape like this, bound by the non-competitive inhibitor, you can see I can increase the substrate concentration as much as I like. It will not bind to this enzyme active site. So this is how the curve looks in non-competitive inhibition. With no inhibitor, the maximum rate here, and this is the maximum rate, that maximum rate is much higher than with non-competitive inhibitor. What this tells us is that the maximum rate of reaction depends, in this case, on the number of active sites that are available, that are free to bind to the substrate. Adding non-competitive inhibitor reduces the number of active sites that can be bound by the substrate, and so with non-competitive inhibitor we have a lower maximum here compared to here. In competitive inhibition then, it is now a bit more easier to understand, having done non-competitive inhibition. So here, let's say that we've got 4 enzyme molecules. Let's say I increase the substrate concentration. More of the active site will be bound by the substrate. Let's now say I have a competitive inhibitor. The competitive inhibitor binds to the active site and it prevents substrate binding. The competitive inhibitor then has a similar shape as a substrate. It's not exactly the same. So let me just there's my competitive inhibitor. Similar shape, but not exactly the same. The inhibitor does not undergo any kind of chemical reaction when it binds to the enzyme active site. It's merely there to block substrate from binding. Okay. Now imagine that if I increase my substrate concentration, because this interaction between inhibitor and active site is reversible, that means that the inhibitor can dissociate, it can fall off the active site. It gives more chance for the substrate to bind. So if I increase the substrate concentration, what it will do, it will displace or kick out the inhibitor and it will bind instead. What this means is that with competitive inhibitor, you can see that you can still reach the maximum rate here, the same rate that I reached with no inhibitor, and in this case I'm keeping the enzyme concentration the same. So in these experiments, the only thing I'm changing is substrate concentration. I'm keeping inhibitor concentration, temperature, enzyme concentration, and other factors the same. So here you can see that although the maximum rate can be reached, for a competitive inhibitor you need a higher concentration of substrate, let's say this concentration, compared to with no inhibitor. To reach the maximum rate. And the reason is we need a higher concentration of substrate to occupy all the enzyme active sites. And that's when the maximum rate is reached. And a higher concentration of substrate because we need to displace the competitive inhibitor from the active site. Okay, so we've looked at some graphs about enzyme rate. How do we go about measuring the enzyme activity? Well, it's important to understand the variables that can affect enzyme activity. They are enzyme concentration. Now, the concentration of enzyme gives an indication of the maximum rate. The higher the enzyme concentration, the more active sites available, so the higher the maximum rate that we can achieve. We can also have substrate concentration. If more substrate bind to active sites, then you're going to get a higher rate of reaction. Temperature. Temperature is important in any reaction, whether it is catalyzed by enzymes or not. Increasing temperature means that there are more frequent collisions between reactants, and this increases the rate of reaction. pH affects the tertiary structure of the enzyme, and thereby it affects the active site shape. So an optimum pH will mean that more enzyme molecules have the correct active site shape to bind to substrate. Type of tissue or species of enzyme that it's from. You might have, let's say for example, you might have an enzyme, let's say isomerase, from different tissues or different species. They're not going to have the same activity. Well, this is something that we can have a variable. Inhibitor concentration. Now, if we look at all these variables, in an experiment, we're going to change only one of them. Let's say the enzyme concentration. and that is our independent variable. The dependent variable is going to be the rate of reaction, what we measure. The other variables need to be controlled. So when we look at enzyme concentration and we change enzyme concentration, the other variables listed need to be kept the same in order to have a fair comparison. Now, it's also important to understand how at A level we go about measuring the rate of enzyme activity. Well, there are generally two methods. Okay, so the first method, you may have looked at this before, is that we have a spotted tile. In the spotted tile, we're going to put iodine, and iodine detects starch. So in a container here, let's say in this flask, I might have a mixture of starch and amylase. I start the timer once I mix them together and they are at a certain pH or a certain temperature. And what I do is every minute or so I will remove a small amount of sample and add it onto the spotted tile with the iodine already present. If starch is still here, then what will happen is it will turn black. At a certain time point, right, the color will change to orange or brown, which is the native color of iodine, which means all the starch has been broken up. It's been hydrolyzed by amylase. And then you stop the reaction because the reaction has ended. So I could do this experiment once under one enzyme concentration, and then I repeat for different enzyme concentrations. Or I might choose another variable to change. And now here it's very difficult to identify a precise endpoint because you might have, in here you might have black color. You might have maybe a lighter black here, and here it may be a mixture of black and orange. And it's very difficult to determine when that color transition occurs. And that's why this is not the best method to use, but it is a method that we use commonly. Another thing you might do is, for example, an enzyme called catalase breaks down hydrogen peroxide, releasing oxygen gas. And this is something that you might have done at GCSE chemistry. You have a container with your reaction mixture. You have a bung with a collecting tube. You then have a trough of water. And here you will have an inverted measuring cylinder which is filled with water. And what will happen is bubbles of gas will bubble up and fill the top, and they will displace the water downwards. So let's say at the top here, right, let's say you filled up to, let's say, 100 centimeters cubed, let's say. And what will happen is the water will be displaced downwards by the gas, and that volume change, right, will allow you to determine the volume of gas that has been evolved over a period of time. So you would time this. Let's say you could measure the volume of gas, let's say, evolved over, let's say, 10 minutes, or you could measure the time taken to evolve, let's say, 20 or 10 centimeters cubed of gas. And then you can work out the rate. And this method is useful. You can do it, instead of using this setup, you can attach the collecting tube to a gas syringe and use the same method. Now, in order to calculate the rate, it's quite important to understand that the rate is the change divided by the time. And this is what we call rate. So the change could be the volume of gas produced divided by the time, or the change could be 1 over time. If we're looking at how long it takes for the color to change, here in this experiment with the amylase, you're going to inverse the time to work out the rate. Okay, now let's look at what happens when we measure pH and temperature changes. So here, let's say we got the rate of reaction, and we've got pH. Okay, let's say we got the rate or reaction, and we've got the temperature. Okay, so here in this experiment here you have like a bell-shaped curve. You can see the highest rate, the optimum activity is at this pH, and this can shift to lower or higher pHs depending on the type of enzyme. For example, let's say a peptidase enzyme, a protease enzyme in the stomach will have an optimum activity at a low pH, acidic pH. So this peak will be here at a lower pH. Now what will happen is, as you come close to the optimum pH, more enzyme molecules will have an active site which is the correct shape. Okay? The other enzyme molecules will be unfolded, and we call this denatured. So at extreme pHs, most or all of the enzyme molecules will be denatured, meaning there's very low or no activity. With temperature then, it starts off similarly, exponential increase, but it's an asymmetric curve, no symmetry. What will happen is that as the temperature increases, the activity will increase because there's more energy. The molecules have more kinetic energy, so they'll react more often. Let's say that's the optimum temperature, highest rate. After that temperature, what will happen is the enzyme will lose its tertiary structure, it will become unstable, and it will denature. And as it denatures, the activity will rapidly drop. There's only a short temperature increase, resulting in zero activity. And let's say at this temperature, most of the enzyme molecules have denatured, only some are active. So how do we calculate rate? Well, whenever we calculate rate, it's always useful to plot a graph. And in this way, we can use a slope or a tangent to calculate a rate. And this is a more accurate method. When we plot the graph, the x-axis is always going to have the unit of time. The y-axis is going to have whatever change we were measuring. So in, let's say, the catalase experiment, it's the volume of gas. Let's say my time's in seconds here. Okay. This could also be, for example, a color change if you're measuring absorbance using colorimeter, or it could be a mass change if you're measuring loss of mass if you're evolving gas. So this can vary, but the overall method to calculate rate is the same. Let's say I've done one experiment where I've used 0.1 moles per decimeters cubed of substrate. So in this experiment, I'm changing the substrate concentration. And I'm then looking, and this is one experiment, so in this graph is going to be data for one experiment at one substrate concentration. And so here, what I will see is, I'll see a slope that gradually gets less steep as I go up. And then slowly, it will level off. Now here, when you have something, a value against time, then the rate, right, is proportional to the slope. If it's a steeper slope, then it's a higher rate. Now here you can see there's no slope at all. It's a horizontal line. There's no rate here, it's zero rate. And here what's happened is that we've run out of substrate, so the reaction has stopped. But as we progress, the volume of gas that accumulates or is released increases quickly, then it slows down because we have reduced substrate concentration, which reduces the rate. Now let's say I do the same experiment, I repeat it, but at a different concentration of substrate. Let's say 0.2 moles per decimeters cubed of substrate. I should expect the rate to be higher here. And indeed it is, so let's say the slope of the curve will be steeper. Okay. Now because it's a higher concentration of substrate, I might also expect a higher volume of gas to be evolved before it levels out. Okay. So more substrate has been converted into product. So here we've got a steeper slope. Now when we plot something like rate against substrate concentration, then what we need to do is we need to work out the rate from each graph, and we'll have 5 or 6 graphs. Each graph will be a single point on this rate versus this variable, which may be substrate concentration or enzyme concentration or temperature or pH. So we need to do a lot of graph drawing, right? And each graph on here, right, will be a single data point on here. So let's look at these two experiments. How do we compare the rate? We need to compare the rate at the same time points to make it a valid comparison. So what we do is we work out the initial rate by drawing a tangent to this line. And we do the same here. And then what we do is we work out the gradient of a straight line, as so, by looking at the change in y, by the change in x. Change in y divided by change in x equals the gradient, which equals the rate in this case. And this is going to be something centimeters cubed per second, because we're dividing centimeters cubed by seconds. Because this value is in centimeters cubed, this value is in seconds, and centimeters cubed per second, whatever that value is. And we do the same for this. But you need to be careful of how you present the units, and you need to look at what is on the y-axis, its unit, and what's on the x-axis and its unit. But this will give you one data point for this initial rate, and you'll plot it on this graph. This will give you a second data point, and you'll plot it, and you will just continue plotting these results. Okay, so here in some exams it also tells you to draw a tangent to work out the rate at a specific time point. So for example, this is the volume of gas, this is the time. For example, if I draw a tangent here to work out the rate at time x, then what I need to know is I need to have the slope of the tangent so that it's equidistant to points either side where it touches the line. So you need to be careful how you draw the tangent. So the distance between this point and our curve, this point and our curve, and these 2 points are equidistant from the center, so it allows us to draw a mean equal tangent. Then we draw a gradient, and we work out delta y over delta x. We work out the slope of that tangent. That gives us an approximate rate for that time. We can also measure a standard rate or a mean rate at this time point. So let's say we start from 0, we go to time, which is 10 seconds. We can work out a mean rate. How do we do that? We just look at the time for 10 seconds, and we look at the volume of gas, let's say that's 10, and then you will do that volume divided by that time. Here we haven't done a slope. Okay, we haven't worked out a gradient. All we've done is, right, we've looked at the 2 time points 0 and 10, and you can see between 0 and 10, right, the volume increases from 0 to 10 here. Okay, so we just divide these two numbers. Now this is a mean of all the individual rates on this slope, which is changing its steepness. Let's go beyond the spec. Firstly, I want to look at how enzymes are regulated through the processes of allosteric regulation and feedback inhibition. Now this is really, really important because enzymes are very important in catalyzing metabolic reactions, and it's very important to control metabolic pathways. And the way this is done is by controlling enzyme activity. So firstly, let's look at something called the TCA cycle, the Krebs cycle, which is part of respiration, and you will learn this in A2. And I'm going to just summarize one important step in this cycle. And this cycle is called a cycle because, firstly, in the first step of the cycle, we use a 4-carbon molecule called oxaloacetate, This is combined with acetyl group from acetyl-CoA or acetyl coenzyme A to get citrate. That citrate is a 6-carbon molecule. It's gradually broken down to oxaloacetate. And in this process, we generate ATP. One intermediate in this cycle is called succinyl-CoA or succinyl coenzyme A. Now, this step, this important step, is catalyzed by citrate synthase. Okay. So oxaloacetate binds, acetyl-CoA binds, and then a chemical reaction is catalyzed by this enzyme to produce citrate. Okay. Now, it's very important in this first step to regulate this enzyme, and this will determine the rate at which this metabolic pathway occurs. So, for example, if I have a high concentration of ATP already in the cell, I do not want to have much of this cycle going on. I want you to slow it down because I don't need very much ATP. So if ATP accumulates, then ATP will bind to citrate synthase at an allosteric site, which is away from the active site. It will change the shape of its active site so it can't bind to its substrates, and then it will inhibit it using non-competitive inhibition. And this inhibition of the first step will slow down subsequent steps because less citrate will be synthesized. And so this will slow down the subsequent steps in the cycle. Another way inhibition occurs is by competitive inhibition. For example, citrate can bind to the active site of citrate synthase and prevent oxaloacetate from binding. So if citrate accumulates, or if there's a lot of citrate, it suggests that other steps further down this pathway are slow or they have stopped. And so it's not very useful to accumulate citrate. It's a waste of energy in the cell. So what happens is accumulation of citrate will inhibit citrate synthase by binding to the active site and prevent oxaloacetate from binding, competitive inhibition. Succinyl-CoA also works in the similar way. It binds to the active site, prevents acetyl-CoA from binding, citrate synthase. Now, when succinyl-CoA inhibits the citrate synthase competitively, this is called feedback inhibition. A product or an intermediate at a later stage in this pathway inhibits an enzyme which comes previously, and that's feedback inhibition. Okay, another important way that enzymes are regulated is by the formation of proenzymes. For example, many digestive enzymes, because they are synthesized inside the cell on ribosomes, if they were synthesized in an active form, they may digest substances in the cell and harm the cell. So protease enzymes, for example, trypsin, is synthesized in its inactive form, and this is called a zymogen. And then when trypsinogen is released into the extracellular environment by glands, into the gut, it is converted into trypsin because it sees another enzyme called endopeptidase. What endopeptidase does, it cleaves a short sequence of polypeptide on trypsinogen, and when you cleave off this short segment, you get trypsin, and the short segment serves to inhibit the activity in trypsinogen. Well, then this trypsin can then go ahead and it can digest proteins. Another example is where chymotrypsinogen, another protease, is converted into an active form called chymotrypsin. And interestingly, trypsin catalyzes this cleavage of a short sequence of the polypeptide to form this active enzyme. Okay. Finally, one important way in which many enzymes are controlled is phosphorylation. Phosphorylation is important in controlling different signaling pathways inside the cell, and you will learn about this in A2. So an important protein which phosphorylates targets It's called protein kinase A. And the word kinase tells you that it will phosphorylate its target by adding a phosphate group. And protein kinase A is activated when glucagon or adrenaline binds to receptors on the cell surface. And then downstream of this protein kinase A, what happens is that it binds to a protein called a phosphorylase kinase. It phosphorylates, this requires ATP, and when you add the phosphate to this phosphorylase kinase, it drastically increases its activity. This phosphorylase kinase then serves to phosphorylate an enzyme called glycogen phosphorylase B, and it greatly increases its activity. When this happens, glycogen phosphorylase goes ahead and it hydrolyzes glycogen to release glucose. And you will be aware that glucagon and adrenaline are signals for stress, and also if there's a low blood glucose concentration, these molecules, these hormones, will stimulate this pathway inside the cell, and it's only when the hormone binds to the receptor on the surface of the cell that this pathway is stimulated. Once the hormone is no longer bound, obviously this phosphate will be removed, and these enzymes will go back into their less active form. And this ensures that glycogen hydrolysis, for example, only occurs when the cell responds to external stimuli, like these hormones binding to cell surface receptors. So this is a very key and very important area in biology, and I would encourage you to read further on this. This concludes this video about enzymes. I hope you found this useful. Please also watch my other videos in the biological molecules section. Many thanks.